I'd like to get started, if everybody's ready. The caterers arrived a little bit late, so we wanted to give folks a moment to get coffee. But I think we should go so that we don't turn the rest of the day behind. I want to spend just one moment doing, for anybody who wasn't here yesterday, a brief revisit of some of what Melanie Marty did to frame the day. Today is the second day and a different focus of yesterday's workshop. So welcome, everybody, to the second day who was here and the new ones. I'm Meg Schwarzman, and I am a research scientist here at UC Berkeley in the Center of Occupational and Environmental Health, and also the Berkeley Center for Green Chemistry. And we've been partnering with OEHA and UCLA to put together these series of short series of workshops aimed at helping OEHA generate or identify the hazard traits and other information that they need to recommend to the Department of Toxic Substances Control. So a quick reminder of the context for this. So under SB 509, the legislation that was passed 2 years ago, OEHA is required, or that establishes the Toxics Information Clearinghouse, and under that law, OEHA is required to, the language from the bill is, evaluate and specify hazard traits, toxicological and environmental endpoints, and other relevant data, leaving it quite open, that should be included in the TIC. And DTSC is building the TIC, but is turning to its sister department, OEHA, to identify what would be the most useful indicators of toxicity to include in that clearinghouse. So the goals for today, yesterday we focused mostly on measures of exposure and indicators of chemical exposure, and the goals for today are to look more at indicators of ecotoxicity. So we're asking the questions, or our speakers will be addressing the questions of what are the emerging methods that we can use to identify ecological hazards? Are there inherent properties of chemicals that indicate potential toxicity? What are the useful screening methods for ecotoxicity that are under development and that could be that could generate data in the midterm, maybe, for inclusion in the Toxics Information Clearinghouse? And finally, how can the information above inform thinking about hazard indicators and potential hazard traits of chemicals? So we have a great lineup of speakers today, and without further delay, I would like to introduce Gary Ankley, who is our first speaker of the day. He's a toxicologist with the US EPA Office of Research and Development, specifically the Mid-Continent Ecology Division, which puts him in Duluth, Minnesota, which I think we'll be able to tell by his accent. Dr. Ankley received a bachelor's in sciences from Fisheries and Wildlife at Michigan State University, and a master's and doctoral degree from the School of Forest Resources at the University of Georgia. So maybe that counterbalanced the Midwest accent. He has been at the Duluth EPA lab for about 20 years, working primarily on the development of test methods for effluents and sediments, including assessment of the effects of endocrine disrupting chemicals on wildlife and the application of genomic and computational toxicology tools to use in ecological risk assessments. He's authored hundreds of papers and related to these topics, and he consults regularly for national and international organizations involved in regulation and risk assessment. So we're grateful that he could be with us today, and I'll turn it over to Gary.
Thank you. It's nice to be here today. Thanks the organizers for inviting me to the conference. As is usually the case, I learn a lot when I come to these kinds of conferences. There was a great lineup of exposure talks yesterday. I hope several of you were able to see that. What I want to do today with my co-speakers is basically cover the world of ecotoxicology, which is not a trivial challenge. As Megan mentioned, I'm from the Duluth EPA lab. This is a charter lab that was formed when the EPA was formed, so we've been there for about 40 years. The charge to the lab was then, as is now, was to develop approaches for dealing with contaminants primarily in aquatic environments. And a lot of the methods and models and approaches that are used throughout the world today in regulatory ecotoxicology actually had their roots in Duluth. What I want to do today is present some thoughts on where we might go in this coming century relative to ecotox testing because of some of the challenges that we face. First, and I'm doing a disservice here because I'm going to cover the history of toxicology, ecotoxicology testing in one slide, which isn't really fair. But I do need this for a little bit of background to kind of frame what comes next. So there are actually quite a lot of toxicity tests out there, both short-term acute and long-term tests, with a whole variety of vertebrate, invertebrate, and plant species that can be and have been used for a variety of purposes in ecotox testing. The data are used for a variety of regulatory purposes, everything from pesticide registration to whole effluent testing, to sediment monitoring, and other types of programs. Although there are quite a lot of test methods available, there's really only a handful of species and phyla that receive a whole lot of testing, and these include cladocerans like Daphnia, chironomids, insects, amphipods. These are all invertebrates. A large fish model that receives a lot of work is the rainbow trout. And then in terms of small fish models, there's three that actually are used quite a lot: Medaka, Zebrafish, and the Fathead Minnow. It kind of depends on what country you're in terms of what receives the most attention. In terms of the types of endpoints collected in these tests, it's primarily what we call apical responses, whole animal responses. And the reason for this is one that's an important distinction between human health and ecological risk assessment, and that's the assessment endpoint. In terms of eco effects, what we're concerned about are effects at the population level, unless we're dealing with endangered species. And so we need to have population relevant information, and that's the kind of information that you get from survival, growth and development, and various measures of reproduction. Less commonly measured in the tests that are conducted for ecotox is behavior, and this whole suite of endpoints is really going to be mostly what I talk about today, as well as several other speakers. Things like histology, biochemistry, molecular responses, things that might be called mechanistic endpoints. Occasionally the data are collected for this, but they're not often used in terms of regulatory decision making. Now, the traditional approach, or the historic approach to ecotox testing, has served a lot of purposes, and it's served them well. We aren't seeing rivers catch on fire anymore. We're seeing effluents that are cleaned up. We do a pretty good job on pesticide regulation, but in reality, the current paradigm isn't meeting all the needs that we need to meet. And this is largely because it's very expensive to run these tests, particularly the chronic and subchronic tests that you really need to do a good job of risk assessment in many instances. And the net result is this: we can only test actually a few chemicals of the universe that we're dealing with, when in fact we'd like to have some data at least for literally thousands of chemicals. This is associated with regulatory programs, both nationally and internationally. I've listed the endocrine disruptor screening program here. I think many of you are familiar with the REACH program in Europe. It looks like TSCA is going to change here pretty soon to reflect something closer to REACH, actually. So we really need data for a lot of different chemicals, which is the challenge that the California group is facing as well. This just isn't possible with our current testing approach that's really focused on apical responses. One of the problems with this focus is it really limits, not understanding how a chemical works, limits our ability to extrapolate to situations that haven't been directly tested. So this emphasis on just apical endpoints limits our ability to extrapolate across chemical structures. It limits our ability to extrapolate from the lab to the field. And it really limits our ability to extrapolate across species, which is a critical one in ecotoxicology, where you're testing just a couple, 3 species perhaps, and trying to understand potential effects on thousands. Now, these kinds of challenges, the need to be able to collect more data with limited resources, are not unique to the area of ecotoxicology. This has been a subject of a lot of conversation and debate in the human health side as well. And this paper report was mentioned yesterday, and it's a 2007 report from NRC that's primarily dealing with human health, but they identify a lot of these challenges. We need more data than we can get, and we need mechanistic data. And they propose some pretty radical paradigm shifts to actually achieve this, and it's been the topic of some heated conversations, but I believe that really the message that they brought out is one that's resonating in the general field of toxicology. Let's see. So these various white papers and strategies that I had up on the last slide, they vary in their context and somewhat in their message, but underlying the message in all of them is the fact that we need to move probably more to predictive approaches to toxicology or mechanistic approaches. We need to utilize better the data we have relative to understanding the basis of extrapolation. We need to develop and validate short-term testing tools, both in vitro and in vivo, that are again focused on mechanism that enable us to predict outside the immediate testing paradigm or environment that we're in. And the goals that we have are to develop a testing program that's like this, that optimizes use of available resources, that results in us being able to make predictions in a reasonable sense of uncertainty, one that's transparent and one that's grounded in established theory, so one that's defensible. Now, stepping back again to ecotoxicology in particular, this sort of approach has a lot of promise in terms of the kinds of things that we should be able to do with a more mechanistic or predictive-based testing paradigm. One involves screening and prioritization. Again, we're dealing with thousands of chemicals that we'd like to have data for. I have to step away a little bit here to see the slides and point at them, so if anyone's having a problem hearing, raise your hand and I'll get closer to the microphone. But basically, we're dealing with thousands of chemicals. It would be nice to be able to go into the chemical universe and use tools to say, these are the ones that we really need to run some longer-term whole animal tests on to understand. Mechanistic information can also help focus the testing. What species should we be testing? What endpoint should we be looking at? Currently, the pesticide registration program has tests that are conducted with lots of different species and lots of different endpoints, and just a little bit of this data are actually used. And so it's not an efficient use of resources for that program. Also, understanding more about mechanisms and pathways can support the issue of cross-species and cross-chemical extrapolation. This final point's not one that I'm going to talk about today. I think maybe others may touch on this, such as Tyrone. But understanding mechanisms and using these types of tools can be very helpful in a diagnostic setting in the field where you're dealing with complex mixtures of chemicals and you're interested in what pathways are being perturbed. So if we have all these wonderful options and approaches and or advantages to using mechanistic tools, why aren't we doing so more in the field? Well, there's both technical and communication challenges, and this slide lists a couple. The first couple bullets here involve technical issues. One, the kinds of endpoints and assays that we associate with mechanistic or predictive toxicology are not what we'd consider routine. The labs out there that do regulatory testing are very good at the types of apical endpoints that have been used, but they're not really all that good usually in terms of histological endpoints, in vivo or in vitro assays, and certainly the whole genomics suite that Chris Vulpe will be talking more about in subsequent presentations. These are more tools that have been considered in the research realm, so they haven't really been applied very much to regulatory situations. Data analysis is complex for these kinds of endpoints. It's not routine or off the shelf. But in my opinion, probably the biggest challenge in terms of bringing mechanistic data to bear in terms of decision making is one of translation. That is, what does it mean when you change one of these diagnostic or molecular endpoints in terms of the types of responses that we're historically focused on, things like survival, growth, and reproduction. And this communication issue is both a science issue and a communication issue relative to the scientists talking to the risk assessors. Now, partly in recognition of this challenge, a couple of years back, a group of senior scientists at the Duluth EPA lab got together to try to come up with some sort of a framework that would aid this sort of translation. And I'm sure that many of you recognize several of the names on this list. We got people here that are involved in predictive toxicology. They're involved in effluent monitoring, sediment monitoring, modeling. And so it's really a diverse group of people who have come at these risk assessment challenges from several perspectives. And we came up with a framework that we think is useful to help provide this translation. I'm going to talk about that a little bit as a basis for the rest of my presentation after this. So the adverse outcome pathway definition is here. Basically, it's a conceptual framework that portrays existing knowledge concerning the linkage between direct molecular initiating event of a chemical and an adverse outcome that's meaningful to risk assessment. So what we're doing is we're tracing basically the events from the initial reaction of the chemical with the biology of the animal or plant and an adverse outcome that's meaningful. This actually builds on the toxicity pathway concept that's presented in the NRC report, but it expands on that concept in several important ways. I think this subsequent slide demonstrates this. What we have here is basically a chemical with specific properties that enable it to interact with different ways with molecular targets. It might be binding to an enzyme, it might be binding to DNA, it might be turning on a receptor. After that, we see a cascade of events across biological levels of organization at the cellular and organ levels that ultimately, if the system's stressed too far, results in adverse effects in the individual. And because we're ecologists, we worry about effects out here as well, things at the population level. The toxicity pathway concept that's presented by NRC is more focused on cellular processes, so it doesn't really incorporate the adverse outcome aspect of this that's important to making the package whole for risk assessment. Now, it's pretty easy to throw that slide up there and say conceptually this is what it looks like, but it's, I find it useful, at least for myself, to actually throw up information that you can relate to. And so what I'm going to do is actually walk through an AOP using real information to hopefully kind of elaborate a little bit for you what it looks like. And in the paper I referenced, there are a number of examples in the paper where we've done this. I'm going to focus on this last one, impaired vitellogenesis. I think most of you know what vitellogenin is. It's referred to as egg yolk protein. It's produced normally in egg-laying vertebrates to support egg production. The way it works is that the lipoprotein is manufactured in the liver through stimulation of the estrogen receptor by endogenous estrogen, typically beta estradiol. It moves from the liver via the plasma to developing oocytes in the ovary, and there it fuels kind of the development and spawning of eggs. So what I want to do here is show an AOP that's related to inhibition of vitellogenesis. And again, here's the conceptual framework, and here are the data that actually line up with this framework. The AOP that I'm going to talk about here involves an enzyme called aromatase. Again, something I think you'll probably hear about later in a talk from Tyrone. But this enzyme is critical in vertebrates in terms of the conversion of the androgen testosterone to estrogen. So it's critical for maintaining appropriate balances of estrogen. It turns out that there are a number of chemicals out there that can inhibit aromatase. These include pharmaceuticals, they include several pesticides. Some of the conazole pesticides are particularly good at this because this is a CYP-based enzyme and they inhibit cytochrome P450. The data in this panel is actually from Fathead Minnow reproduction test, and it's with a chemical called fadrazole. This is a pharmaceutical that's a specific inhibitor of aromatase. It was developed to treat breast cancer, actually, estrogen-sensitive breast cancer. And what we did is we did a 21-day exposure of fathead minnows to this chemical through the water. And here are some of the data that come from the experiment. I'll have to tell you what's on these axes because you really can't see them, but this is estradiol levels at different test concentrations of the chemical. Here's the controls. And what you see is a nice dose-dependent decrease in circulating estradiol levels in the fish. This is what we expected to see. This bottom panel is the consequences of this in terms of vitellogenin production. Again, here's the controls. With the lack of stimulation of the estrogen receptor, what you see is a dose-dependent decrease in the amount of vitellogenin that's in the fish, in the plasma. If you step up one biological level of organization further that corresponds to this, this is what happens at the cellular organ level. This top panel is a, sorry for this pointer, it's not working quite right. But this top panel is from a control animal. This dark staining material is actually deposited vitellogenin. This bottom panel is from an animal that's been exposed to fadrazole. You're seeing atritic oocytes and basically no deposition of vitellogenin, which is consistent with what you'd expect to see. What's really important here and critical is this last panel here. And this is the ability of the fish to produce eggs. These lines up to this point here are basically a pre-test evaluation of spawning cumulative fecundity. This is where the fadrazole is turned on, and you can see it has a dramatic effect on the ability of the animals to produce viable eggs, basically shutting down egg production. So what you see here is basically the path all the way from a molecular initiating event through various processes to actually an adverse outcome at the individual level. Well, you know, this is a simplistic depiction. We know that pathways are more elaborate than this. They branch. You can have one initiating event producing multiple adverse outcomes. You can have several initiating events producing the same adverse outcome. But you can build on this framework and use this. And this is an example of that. This depicts, and I'm not going to go into a lot of detail here, but this depicts 3 different ways that you can affect vitellogenin production, 3 different molecular initiating events that all produce the same sort of effect in terms of egg production. I talked about aromatase inhibition here with the fadrazole. You can also reduce vitellogenin through antagonism of the estrogen receptor. Essentially, you block the estradiol from stimulating receptor. There are chemicals out there that do that. You can also decrease vitellogenin production with androgen receptor agonists. The way we think these work is through feedback inhibition of steroidogenesis. So essentially, you're decreasing the estradiol signal. And so these 3 different molecular initiating events all converge on this one node here of vitellogenin, which then we can relate to adverse outcomes up here in the individual and in the population. So where does this get us besides some nice graphics? Well, it really enables insights as to how we can do risk assessments, both prospective and diagnostic risk assessments. Prospective assessments being those that for chemicals that we haven't released yet, hopefully, to the environment, and diagnostic would be more what we'd consider to be field work. In terms of prospective assessments, what this does is it provides us with the types of insights we need to develop what are called tox pathway assays. Short-term in vitro or in vivo assays that are focused on mechanisms that we know have the potential to result in adverse outcomes. So in this instance, we can clearly define why we'd want to be measuring antagonism of the estrogen receptor, activation of the androgen receptor, inhibition of aromatase. Intuitively, we feel like we ought to know this, but this actually anchors those sorts of endpoints. In terms of a field setting, there's a lot of emphasis out there on collecting what we call biomarker information from the field, and one of the things that people like to look at are circulating steroid levels and circulating levels of vitellogenin. And again, we know that this is indicative of some perturbation in the endocrine axis of the fish, but by having these pathways laid out and understood, we can actually relate these sorts of biomarkers to what we would consider to be adverse outcomes both in individuals and populations. So with that kind of background in terms of adverse outcome pathways, what I'd like to do for the rest of the talk is specifically present some of the tools and approaches that are available out there that I feel that in the short, moderate, or maybe even long term that could be brought to bear on some of the questions and issues, challenges that we face in ecotoxicology. First, I want to talk about accessing data that actually are available and using these effectively. I want to talk also about leveraging data that, although not developed for ecotox purposes per se, can be brought to bear on the situation. I'm going to touch on computational or QSAR models for predicting adverse effects. And then I'm going to talk a little bit about in vitro and in vivo assay systems that can be used to sort out pathways or these adverse outcome pathways that provide both mechanistic data and indications of potential adverse effects. First, I want to talk about utilizing data effectively that we already have. There actually is a surprising amount of information out there that is ecotox oriented. And some years ago, some folks at the Duluth lab recognized this, and with a lot of foresight, have put a lot of effort and resources into a database that's known by various acronyms, the most recent of which is Ecotox. And what this is information that's been compiled from the literature by people that are experts in assessing this information and inserted in a database. And this is data for aquatic species and more recently as terrestrial species as well. So there's some level of QA that goes into this. They're not assembling garbage. If you're going to use it, you actually need to go get the original papers. But this is by and large stuff that has received some level of QA in addition to actually obtaining the information. Importantly, this is a publicly available resource, and it can be very useful. It's searchable in different ways. It's updated every quarter. I think as of the latest update, there's about 750,000 records present in Ecotox from over 30,000 references, and I think it captures about 9,000 species and 9,000 different chemicals. That's clearly not the whole chemical universe, but I've found the database in many instances very useful in locating data for chemicals that I thought maybe weren't even tested, but also you can find related chemicals that have been tested, and this can give you insights as to what you may be dealing with unknown chemicals. So it's a nice compilation of existing information that I think can be tapped into to address the sorts of challenges that California is facing relative to populating your database as a first step. What I want to touch on next is leveraging information that, although it was not necessarily developed for ecotox purposes, still could be used at least for a screening level risk assessment. And for my example here, I'm going to touch on chemicals known as pharmaceuticals. I think most of us are familiar with these. They've been in the news quite a lot. Basically, the story here is that we're increasingly detecting a lot of these in the environment. Part of it is better analytical techniques, but part of it is greater use of these chemicals as well in human populations. Here we're talking about potentially thousands of chemicals when you consider parents and metabolites. There's a lot of concern for possible human health issues in terms of drinking water. And we have documented effects of pharmaceuticals in terms of effects on wildlife, feminized fish associated with EE2, diclofenac effects on vulture populations are just a couple examples that are pretty well documented and out there. These chemicals, and I mentioned this yesterday as part of our exposure discussion, are not generally all that persistent in the environment. There's a few that are, but most of them aren't. But they have a property called pseudopersistence that's rather important that we need to consider when we're looking at these chemicals, because people are constantly using these. They're constantly introduced to the environment, and in effluent-dominated systems in particular, the exposure would look like a persistent exposure to the animals that are in this environment. A lot of these pharmaceuticals target what we would call conserved pathways. So the target species, us, or in the case of veterinary drugs, certain livestock, a lot of these pathways are actually in a lot of non-target species that we could be concerned about. An important point here is they tend not to be acutely toxic. So we have acute tox data for a lot of pharmaceuticals, but most of it's basically useless because they're targeted on pathways that are more associated with chronic effects. And as a result, they can be very potent. One way of measuring this potency or assessing this potency that's been used quite a lot in ecotox is what's called the ACR ratio, acute to chronic ratio. This is basically the amount of chemical that's necessary to cause acute effects divided by the amount that would cause a chronic response, such as a reproductive response. A lot of pharmaceuticals have ACR ratios greater than 1,000. This is really quite unusual for our field. Typically, chemicals we work with have ACR ratios on the order of 10 to 20, very seldom more than 100. And so we're dealing with really kind of a unique situation here with pharmaceuticals. The challenge here, though, is that there are basically, because of various regulatory issues that I really can't get into too much today, there is almost no data that are focused directly on exposure and effects issues that's relevant to ecotox. And I think actually Phil identified this yesterday as part of his presentation as well. But this is where the leveraging data comes in. There is a lot of information that's collected in conjunction with development of these pharmaceuticals and their safety assessment. It's primarily human-oriented that can be leveraged to at least a screening level assessment of their risk in the environment. And this type of information includes basic physical chemical property information, things like KOW, solubility, vapor pressure, some pharmacokinetic parameters that are related to therapeutic issues in humans, things like absorption, half-life in the system, biological targets and pathways for the drugs, and effective dose, which is a measure essentially of potency. Now, there's been a number of meetings, some held by CTAC. There's been a White House level working group that I was involved in that have talked about how you could actually leverage this sort of information in a systematic way to get at the issues that we're concerned about relative to the ecological risk of these chemicals. And basically, I know the feeling out there is that you actually can directly address these sorts of issues with the kind of information that's available, particularly for human pharmaceuticals, which likely would occur in the environment. What are metabolites or degradates that could occur that could be stable enough to be of concern? Are the biological targets and pathways that are affected by these chemicals relevant to ecological processes that we may be concerned about? What is the degree of conservation of these pathways across species? And finally, how potent are the chemicals? I could spend a whole talk kind of going into these issues, but I have just a couple slides I wanted to put up there because I think they help illustrate how you can actually address some of these points. Here's a figure from a paper from a lab in Sweden, Joaquin Larsson, that was published a couple years ago in Environmental Science and Technology. And what they sought to do here was basically look at the conservation of these targets across species for animals, for organisms whose genomes had been sequenced. So humans are here on the left. This is the target species, and here are the target biological molecules roughly arranged by class here. And what we have walking across here, I think it's a bit of a misnomer to say that we're going down levels of biological complexity, but here are the vertebrates, here are the invertebrates, and here are bacteria. And what it shows is the biological sequence conservation of the drug targets in these species. And this is pretty important information because it tells us which chemicals should we be concerned about in terms of where they might affect the environment. So we aren't going to worry about receptor-based molecules, probably in most invertebrates or bacteria. We may be worried about some conserved processes relative to microbial function, though. So this type of information, if collected appropriately, enables us to identify which pharmaceuticals maybe we ought to be most concerned about and how we ought to test them. And I can't go into more detail, but we're doing a finer analysis of this now in our lab to try to actually work with species for which there aren't full genome information, and we're making some real nice progress in this area. Another, I think, relevant example from the recent literature is a paper from Brian Brooks' lab in Baylor, Brian's been worried about pharmaceutical issues in the environment for a number of years and done some very nice work. And it shows again how you can leverage some data from human health in terms of eco-health. And what they actually did was they took some information on the mammalian acute toxicity. This is a concentration of a drug that causes adverse effects relative to its therapeutic levels, the ATR ratio, if you will, in humans. And for the data that they had they plotted it against the ACR ratio of several chemicals in fish. And again, this is a nice measure of potency. And what they found was not surprisingly, a very nice relationship here. So here we can actually use this type of data from the human health side to help target those chemicals that are most likely to be potent in non-target species. So what I want to do now is actually move from considering existing data and leveraging this data to some of the predictive approaches that are available out there in terms of dealing with chemical data sets. And here I'm going to pick on an example that I think is well known to people, but it involves prediction of narcosis toxicity. Narcosis toxicity, it's also known as baseline toxicity, is the toxicity that a chemical will exert if it doesn't have a more specific mode of action. So basically, any chemical, if you can get the dose high enough, will kill an animal at some point. The phenomenon here looks to be involving hydrophobic interactions of the chemical with cellular membranes. So at some critical cellular membrane level of the chemical, it will disrupt processes inherent to that membrane and result in essentially behavioral effects and ultimately death in exposed species. This is important because it's estimated that probably up to 60% of the industrial chemicals that we deal with out there actually have this mechanism of action as its primary pathway. So what I want to do now is talk about narcosis relative to this AOP construct that I described earlier. Narcosis is an interesting example in that we know something about the molecular initiating event. It's essentially movement of the chemical into cellular membranes that somehow affects these membranes, for example, affecting fluidity of the membranes, and ultimately resulting in adverse outcomes, loss of equilibrium. In this case, we're talking about fish. Well, people too. If you drink alcohol, you can see how that happens, and ultimately mortality. But we don't really know a whole lot about what happens in between relative to this pathway. What are all these things? And so this is in contrast to the vitellogenin example that I presented earlier, where we have a pretty good idea what's in these boxes. But the point here is that if we can describe this relationship well, we might not need all this information in order to make predictions up here. And it turns out that there is actually a parameter that describes quite well the ability of a chemical to interact with cellular membranes. And there was quite a lot of discussion of this parameter yesterday, the octanol water partition coefficient. So this is a very useful property, not just for environmental fate, but for predicting toxicity. And in fact, if you consider, this is data that was collected for many years at the Duluth lab, where tests with a whole variety of narcosis chemicals were conducted in 4-day assays with the Fathead minnow. And here you see a kind of a summary plot of the data where the log KOW is regressed or plotted against the 96-hour LC50 for the fish, and you see a very strong relationship here. This relationship has actually been used as the basis for developing a QSAR that's used actually throughout the world. It's used in the US, it's in the EPI suite that was referred to yesterday. It's used in OECD in their toolbox as a basis for discerning narcosis toxicity and making predictions of this in fish. And it turns out one of the nice things about this is it's very nice in terms of extrapolating, at least across aquatic species. There's not a lot of differences in terms of inherent species sensitivity to these chemicals. And so here in terms of the AOP, I think we're able to say that if we understand key properties of the chemical relative to the molecular initiating event, we can go right to the adverse outcome and make these sorts of predictions. So we don't need to spend lots of resources understanding this to actually apply this tool. What I want to do now is switch to those chemicals for which we don't have very much information for, that we may need to collect information for. So we don't have databases to consult, we don't have data to leverage, we don't have models that are appropriate, QSAR models that are appropriate for predictions, and that's the case for most reactive chemicals. Most of the QSARs out there for reactive chemicals are not well enough refined to actually use for risk assessment. So we still have a lot of unknown chemicals to deal with. And what I'm going to talk about here relative to doing that involves in vitro and in vivo tests where you develop suites of responses or fingerprints that you can then compare unknown chemicals to in terms of identifying what pathways they might affect. Some of you have probably heard about this program. It's called ToxCast. This is actually an effort that's resident in the Office of Research and Development of EPA. It's coordinated through the National Center for Computational Toxicology. And what they're seeking to do is take a large suite of in vitro tools and develop fingerprints based on chemicals with known pathways or known mechanisms of action, and then use this as a basis for comparison of unknown chemicals so that you have this suite of known chemicals, you know how they work, you put in an unknown chemical and you can say, aha, this pathway is going to be perturbed. This effort's been primarily oriented towards human health, but a lot of the pathways that they're concerned with are actually conserved, and so they actually are applicable to some of the challenges we might face on the ecological side. Even if I had the expertise, I don't have the time to go into the suite of assays that they're using, but there are things that many of you who have worked in the lab would recognize, things like inhibition of receptor binding and inhibition of specific enzymes, binding of DNA, effects on energy metabolism, and all. There's about 300 different endpoints associated with the assay suite that they're using. And they're approaching this project in a phased fashion. The first phase of their testing involves a fairly broad set of chemicals, about 300 chemicals, for which they have a pretty good idea what pathways they light up. And so this is sort of establishing the baseline data from which that they would make interpretations. The NCCT now is involved in what's called phase 2 of the studies. They're expanding this universe of chemicals, I think, to about 700 of both knowns and unknowns. If there's an interest in this in the audience, I've put the website up here. You can go to that website, get more background on this program. They have a couple papers out there now on it as well. Now finally, what I want to do is talk a little bit about in vivo systems that enable you to kind of sort out pathways. And there's a lot of examples that I could pick on here, but I'm going to pick on one that we've been involved in because I know about it. This is actually a project that's been ongoing now for about 5 years and involves a relatively large group of scientists from all over the US, and it's a very strong consortium of molecular biologists, toxicologists, analytical chemists, and systems and network and population modelers all working together to kind of achieve the objectives listed on this slide. So I don't need to read these to you, but the goal is to actually sort out pathways within the HPG axis and understand the relationships between some of the mechanistic and diagnostic responses that you can measure at the molecular and biological level and adverse outcomes at the individual and population level. In terms of what we're working on, here's a simplified depiction of the HPG axis. So this is what we're talking about when we talk about endocrine disruption. What we all think about, or many of us think about, however, is just this one little piece. This is the ER, the estrogen receptor. And so when people talk about EDCs, they talk about estrogens, but in reality, we're dealing with a lot of different points that can be disrupted in this axis, ranging all the way from interactions with the GABA receptors in the brain, the dopamine receptor, various aspects of metabolism and synthesis of steroids, binding to the androgen receptor, agonists, antagonists. And so this isn't just an estrogenic pathway. There's a lot of different ways that we can disrupt this. And so what we're doing in this project is actually using probe chemicals with relatively established mechanisms of action or molecular initiating events to kind of trace through what happens in our fish models. And the species we're using for this are a couple. There's the zebrafish, which is rather a nice species in the context that its genome has been sequenced, so this helps aid us in the interpretation of some of the omic data that we collect from the studies. And we're also working with the fathead minnow, which is used extensively for regulatory activity in North America. It also is ecologically relevant, and it turns out that actually the genomic tools that are available for this species are really emerging as well. And soon I expect they'll be actually parallel to the zebrafish. For example, we're currently working with a consortium of folks to sequence the fathead minnow genome. So the exposures that we're conducting with these probe chemicals are experiments of various lengths where the chemicals, the fish are exposed to chemicals via the water using state-of-the-art facilities where we're using flow-through systems, measured chemical concentrations. So these are high-quality experiments, and the data from them are high-quality, suitable for modeling. And in terms of the suite of endpoints that we're looking at, there's a lot of information that's coming out of the experiments with each chemical. It starts with the sorts of apical responses that are, again, meaningful to risk assessment, but it proceeds through a number of endpoints that are indicative of function of the HPG axis, things like steroid levels, gonad histopathology, and it also includes a number of what we call an omic responses, transcriptomics, primarily with microarrays, proteomics, and metabolomic responses. I'm not going to talk too much about the omics component of the study, but I think you can envision why we need 12 labs from throughout the country involved in this sort of work in order to make this coverage. Now again, as I was just reminded now with the 5, I don't have time to get into actual data from this project, but I want to just give you a flavor of why it's being done the way it's being done. And I picked this chemical out because I like it, but it's not anything special. This is a synthetic androgen that actually has some environmental relevance. It's 17-beta-trembolone. I'm not going to get into why it's relevant, but I picked out the slide to illustrate that we're collecting apical information that's actually useful for anchoring all these molecular endpoints. So you have to anchor these molecular endpoints in something that's meaningful if you're actually going to say they are going to work for risk assessment. In this case, you can see that trembolone is actually a very potent reproductive toxicant. It affects egg production in the fish at nanogram per liter concentrations of the chemical in the water. And then we're able to relate this sort of apical information to the molecular diagnostic measures that we make in the experiments. And here are some of the HPG axis genes that we know are important to this based on an understanding of the system. These are measured via PCR. Again, I can't go into what all of these do, but we're able to measure these genes, relate them to what we see with regard to an androgen exposure, and then relate these biomarker or biochemical responses to an apical outcome, and I'll show an example of that here. This actually traces back to the AOP vitellogenesis example I presented earlier in the talk, but what we're able here, through understanding how these pathways work, is essentially relate this biomarker, vitellogenin in females, to their ability to produce eggs. And this is data from chemicals that have 3 different molecular initiating events. And so you can see why this is a very powerful tool to be able to relate the systems approach to adverse outcomes. And a neat, really slick thing that you can do with this then, since you have relative fecundity, is you can actually go into population modeling. Population models require some sort of measure of survival and ability of the animals to produce eggs. And so here's an example of us applying this information to a population model. Here's a baseline or control population. And here are the effects of different depressions in vitellogenin on population status. So here's a 25% reduction in vitellogenin. And you can see over time, a population of fish experiencing the stressor would go to less than 50% of carrying capacity. and about a 50% reduction takes them very close to extinction. So here we've been able to take actually mechanistic data or biomarker data and relate that data to a population relevant response. Something else that's coming out of the project is basically providing us the information to develop short-term assays that would enable us to actually fingerprint chemicals with different mechanisms of action or different pathways within the HPG axis. These are laid out here. These are the test chemicals. And here are a number of the gene expression PCR endpoints that we've looked at, as well as a couple other measures. And what you can essentially do is develop heat maps that would enable you to discern between different pathways within the HPG axis. And so you could take an unknown chemical, probably run a short-term exposure, say a 4-day assay, look at a number of genes and responses. Some of these genes would be amenable to, say, high-throughput measurements or high-throughput type measurements in terms of automation, and make assessments or predictions as to what pathways you're affecting. One of the really beautiful things about this is because we've done these longer-term studies, we can now relate results from a short-term screening assay to something that's meaningful to people in terms of risk assessment. So to conclude what I have here, generation of data to meet current needs for risk assessment on the ecological side cannot be wholly achieved with how we're currently doing things. That is apical long-term tests. We simply can't afford to do it. We have too much to test. We need to know more than we know, can know. But some of the newer tools out there in terms of bioinformatics, genomics, and in particular systems biology enable us to bring information to bear that provide a mechanistic understanding that enable us to, I think, essentially bypass this shortcoming if we focus our efforts. But this isn't going to be easy in terms of research or regulatory implementation. Use of these types of predictive tools requires rigorous, validated methods for collecting the information and interpreting the information, and also a transparent linkage of the mechanistic data to adverse outcomes. And I think here is where the AOP concept can be very useful. So I think that concludes pretty much what I have.
Questions, and there will be more time for discussion later.
Well, thank you for a very nice presentation. I have one quick question. In this data that you presented, some of this data sometimes is evaluated using clean water in the lab, but some chemicals in the field sometimes react with dissolved organic carbon. Is there any data to see the effect of this organic carbon and how you can extrapolate from laboratory conditions to field? And the second part of the question is there data for the non-observed effect concentration so people can compare that to the environmental concentration and do a risk assessment, PEC, PNEC analysis?
I think your first question involves... Well, if Chris will let me, I will. Well, I think your first question involves one of exposure. You're talking about organic carbon partitioning, and really that's not the goal of what we're trying to do. I think that was more a topic of things that were covered yesterday, and certainly to do the lab to field extrapolation of exposure, you need to understand the partitioning of the chemical in the field. And an important thing, obviously, there for organic chemicals is they're partitioning the organic carbon. And I think Phil Howard can speak to that much better than I, but that's part of the equation. It's really not what we were focused on. I think your second question involved whether the chemicals we were working with, whether the concentrations we were working with are relevant to the field. And some of them are, some of them aren't. But really the purpose of what we're trying to do is a proof of concept in identifying ecologically relevant pathways, not always using ecologically relevant chemicals. Some are EE2, Trembolone, a couple of the other chemicals are ecologically relevant. But what's key to what we're trying to do is to affect pathways that we know are ecologically relevant. So in that sense, what you're asking isn't directly applicable to what we're trying to achieve, but I appreciate what you're saying.
Susan Kegley, Pesticide Research Institute. I guess just the real quick clarification question on the Fathead Minnows slide with the log KOW and the effects. How do you know that's just not due to less being water soluble? Because your log KOW gets higher, you have a less polar substance, it's not dissolving in water. How do you separate that effect from this binding to cells effect?
Well, you're plotting a chemical property versus LC50. So you've achieved a water concentration that's sufficient to kill the animals.
So Chris Vulpe is an associate professor in nutritional science and toxicology in the College of Natural Resources at UC Berkeley. He received his MD from the University of California, San Francisco, and a PhD also from UCSF in genetics. His group is currently working on 3 primary issues. One is the metabolism of eukaryotic copper and iron, or copper and iron metabolism in eukaryotes, genomic approaches to identify the toxicity pathways that are conserved among some species, and also ecotoxicogenomics, which I think is going to be the focus of today's talk.
Okay, thanks very much for inviting me, and it was a long walk. And I just wanted to Meg asked me to talk a little bit about some of the approaches that we're using in our lab, and maybe also talking about, in general, about how a lot of the methods in omics and other screening tools could be applied to the issues that are facing California with the green chemistry initiative. So you're well aware of the problem, which has been alluded to many times, is that we have just too many chemicals to efficiently or effectively test, and the problem is an overwhelming amount of cost involved with the current testing systems. So how do we deal with this? Well, I think this has already been alluded to, but one of the issues with ecotox, which is a little different than standard tox, is that you're dealing with really an ecosystem where there are multiple different species. So your problems in some way are magnified in that not only are you trying to predict toxic effects just for one species, humans, you're trying to predict toxicity for a whole series of different organisms. Ultimately, your goal is to predict what's the toxicity to an organism in the field. We're not able to effectively do that currently, so we use a series of eco-indicator species, which are sort of representative species at each level in an ecosystem. So in this example here of an aquatic ecosystem, you have a primary producer, and a common algae is often used as an indicator species. A consumer such as Daphnia, a secondary consumer such as a small fish, and a larger fish. So you use a series of these eco-indicator species to help you predict what's the likely toxicity to a chemical. The current approaches, as Dr. Ankley referred to, are what I call basically the kill 'em and count 'em approach, which is that you abuse your favorite test organism and ask how many of them have passed away after some period of time. And I guess part of the issues with this is that it doesn't really tell you why the animals died. It doesn't give you any insight into that. It just, they're dead. It doesn't really tell you what killed them. And I think one of the particular issues is it doesn't really identify sublethal endpoints. So it's not going to identify things like effects on immunity or effects on prey avoidance, or a lot of things which could have important implications for the survival of that organism in the environment. And it's bloody expensive. So I think that's one of the major problems with it. Similarly with field ecotox, a lot of them are based on a similar approach where you basically go in and evaluate a site and look for basically, you can't count what's already dead, so you count what's there and you sort of extrapolate to what used to be there or you thought was there. So as Dr. Ankley again mentioned, basically what's currently done is that risk assessors try to take all of this information from a whole battery of animal tests, and they basically, for all the chemicals which are tested, a sort of indiscriminate number of assays are done on a whole wide variety of species, and the vast majority of this information is not actually even utilized. And so you have a whole lot of data for a lot of species on very few chemicals. And I guess the problem with this is that you don't really have any sense of, well, which of this information should I use, which of the information is actually helpful for risk assessment. And the vast majority of the information, as Dr. Ankley mentioned, isn't even used. So you waste a lot of time and resources. So the question is, can you come up with a better way to do this, to try to more efficiently use the limited resources that are out there and really identify the endpoints that you're interested in. And as talked about by several speakers, there is the growing realization in the toxicological community that we need to develop new approaches to assess this. And these are things that you've seen before, but I think the idea is to focus on toxicity pathways, the application of toxicogenomics or other approaches to sort of understand mode of action, to develop high throughput and what are often also called now high content screening approaches, where you are assaying a lot of different endpoints at the same time, and also coming up with in silico or computational measurements, computational approaches to allow you to predict whether a chemical is likely to be toxic or not. And Dr. Ankley talked about the KOW. And I'm going to focus today a little bit on the three first ones. I'm not really going to talk about the in silico approach, and try to illustrate a little bit about how these approaches could be used for screening in ecotoxicology. So toxicity pathways, as originally defined, suggested that you go from, in a normal individual, you have sort of the biologic stressors or inputs, which then ultimately lead in normal function. But at low doses, you can get perturbations of a toxicant. You can get perturbations which affect the organism, but the vast majority of these perturbations are going to be modulated by the normal homeostatic process of the organism, and they're going to adapt to that situation, and that's sort of the normal cellular and organismal response to stress. When you get into doses which lead to problems, then that can ultimately end up in causing morbidity or mortality. And so this is sort of the concept of toxicity pathways, and trying to understand all the steps along in this pathway. Currently, we really don't have information. We have some information on the initial steps, and we often have information on the final steps, but we don't really have an understanding of what's happening in between. So again, I think this is interesting because we have another example of a toxicity pathway, and this was a well-known one, the nonpolar narcosis that Dr. Ankley talked about. And this is just showing sort of how we would think of such a pathway going from a xenobiotic leading to defects in the membrane, which would then ultimately result in organ problems, including respiration and circulation, and ultimately leading in cell and organismal death. And so the idea here is that we can potentially come up, by understanding this process, we can come up with ways to assay the early endpoints, the molecular targets, without having to necessarily carry out the assays on the whole animal. So what is the potential advantage of the toxicity and adverse outcome pathways? Well, this was sort of my take on how could this information be used to help the Green Chemistry Initiative. And my thought is that it can probably enable a more comprehensive, high throughput, and cost effective. And what I mean by comprehensive is that currently we choose endpoints not necessarily based on any sort of real understanding of what all the possible endpoints are that are out there. We've chosen a few that are of particular interest to us, death, reproduction, but we miss a lot of endpoints such as immunity, prey avoidance, vision. There's a whole variety of things that we don't assay for. And so if we use this more comprehensive approach, then we can try to define, well, what are the pathways that are out there? What should be really concerned for? And direct us to what we should assay. And we're not going to miss the important endpoints. We're not going to be in the situation as we were with endocrine disruption that we'll suddenly realize, oh, well, we weren't looking for this. We didn't see it. And so if we can come up with a way to identify the endpoints we're interested in, then we can make sure that we ask for them. High throughput. Well, I think this is based on the idea that once we have an understanding of these adverse outcome pathways, we can, and as Dr. Ankley sort of talked about, then you can identify the early events. And you can come up with assays which will screen for those early events. So you don't have to do all of those you don't have to do all of the assays on the whole fish. You can do it on a small on a series of assays, and you can use high throughput approaches to be able to address that in cellular systems, and I'll talk a little bit about that. Currently, though, our systems are largely not really driven by the adverse outcome pathways. They're driven by what we happen to have around in the lab. And so I think as I'll talk later, I think there needs to be a real effort to define the questions we want to ask and then develop the appropriate assay systems to ask those questions. And finally, I think that this can be a more cost-effective approach for two reasons. One, we can choose the assays that we want to study based on the adverse outcome pathways that we would like to assess. And it'll also allow basically a tiered response. So we won't have to do all of these assays on all of these fish or Daphnia or whatever. We can use an approach where we screen for adverse effects using these high throughput methods and then carry out targeted analysis on chemicals of concern or on endpoints that were identified by that thing, by the in vitro assays. So it allows us to narrow our search and focus on the endpoints that we're interested in. So this is just a sort of an example here of that, where you would take an existing data, which was Dr. Ankley referred to, and you can use these tools, including existing data, the in silico data, and you can carry out what I would call a prioritization screening. And once you've done that prioritization screening, then you can carry out confirmatory tests to determine whether it actually is leading to an adverse outcome or lethality in a whole animal system. And this was referenced here from this was not a new idea. This came from the McKim conference on predictive toxicology back in 2007. Okay, so sort of gave an introduction on the toxicity pathways and the adverse outcome pathways. And now I was going to turn to two separate issues, which was the application of toxicogenomics or genomic technologies and how they can be used in this approach to screening chemicals. And then I'll talk about the use of this high throughput or high content screening approaches. So ecotoxicogenomics, for the people who are not familiar with it, is basically just a combination of the tools which you're probably familiar with now in genomics, which is measuring gene expression level or protein expression levels or metabolite levels, and trying to meld that with our standard ecotoxicology endpoints, which are often some sort of toxicity testing, bioavailability, or ultimately with understanding and assessing what's happening to a community or a population of organisms. And this is a relatively new field and has really built on the efforts in the mammalian community to use genomics for the assessment of toxicology. And the basic concept is that an organism is going to respond to its environment by turning on particular genes, pathways, proteins, turn off particular pathways, and it's going to respond to its environment. And that environmental response is something you can measure. And by measuring that response, you can get insight into what the organism is experiencing. So you're basically, it's a holistic approach in that you're letting the organism tell you how it's feeling. And as it's going through its traumatic event, it is gathering information that it's going to pass on to you, and your job then is to interpret that information and try to understand what is that organism seeing. And there are many ways to measure this, and I'm going to focus primarily on transcriptomics, or the ways that gene expression information can help you understand things. The general approach with this is often relatively simplistic, in that you're generally taking organisms that are not abused, and you're comparing them to organisms that have been abused, and you ask, well, how do they compare? What's happening in the exposed individual that's not happening in the control, and vice versa? And you use a variety of different technologies to do that, and one of these is the microarrays, where you are looking at a whole series of genes. And what it allows you to do, as shown here, is that you can use an array, and each one of these would represent a different gene. And so you can look at, and these arrays can have tens of thousands or hundreds of thousands of genes on them, and you can identify what's happening to every gene at the same time. And so this is a sort of a massively parallel way to ask what's happening to the expression of genes in these organisms. So what can you do with this information? Well, there's a lot of approaches, a lot of utility to these ecotoxicogenomics, and I'm going to focus primarily on the third point, which is that it could potentially be an approach to help you predict adverse consequence on a population, and also to, I think more appropriate for this thing, enable prioritization of chemical toxicants. I also think that it's important in the identification of mode of action, and that information could be useful also in the establishment of adverse outcome pathways. So to turn just to a little bit to our work, and I think one of the questions was, well, if we're going to be able to use this for screening of chemicals and for prioritization and for understanding mechanism of action, one of the key questions was, well, do organisms actually respond in a specific way to chemicals? And this may seem sort of obvious now in retrospect, but when we started this work, there was a feeling in the field that a lot of organisms are going to respond in very similar ways, that they're going to all basically have very similar stress responses, and that there's not going to be any utility to looking at all of these different chemical responses because they're all going to respond in the same way. So we started a simple project to ask that question. Well, do Daphnia respond differently to different chemicals? And this was a project that started by Helen Poynton, who's now at the US EPA. And what she found was that, in fact, yes, if you expose these suckers to different chemicals, they respond in a very unique and reproducibly different way. You can identify the specific pathways that they respond to, and you can also identify individual sets of genes that respond and are confirmed by the other approaches such as quantitative PCR. And many of the markers that we could identify actually showed a dose response. And so you had specific genes that were differentially expressed, and some of these genes actually would respond with a different, with a response to the dose of the chemical. So you could get information both about specificity of the response and the dose, and both response both in the acute and chronic exposures. And one of the questions we had, well, what does this mean? Does this have any utility for the organism? And one of the things we were surprised with this example with zinc was that it turned on a whole series of chitinase proteins, turned on a whole bunch of chitinase genes, and we were really surprised by this because there was no previously sort of been described. And so when we looked at this, we then actually measured chitinase activity and found that chitinase activity was actually decreased in these organisms, and that actually correlated with a decline in reproduction. And so what was exciting for us was that we could, by doing a 24-hour assay, we would be able to get insight into what would potentially be happening after 21 days. So there was a the first sort of indication that these gene expression pathways actually are linked to downstream effects, such as adverse outcomes. So does this hold for additional chemicals? And so we started a collaboration with another group at the U.S. Army Corps of Engineers and looked at a variety of additional chemicals. And in fact, we found that, again, there were very similar, very distinct expression profiles. What was interesting was that, that closely related chemicals, even though they were similar in their expression patterns, they had very distinct expression patterns. What you're seeing here is a heat map, which is showing on this axis the various genes. On this axis are the chemicals, and these green and red are representative of differential expression. So a green would indicate a gene which was downregulated, and a red would indicate upregulated. And so what you can see is that each chemical has a distinct profile, and closely related chemicals actually have related expression profiles. And you can identify series of genes which are specific for each individual chemical. So you can come up with basically a diagnostic tool to allow you to identify whether an organism has been exposed to a particular chemical. Recently, we've now expanded this now to look at a series of 36 chemicals, which are the EPA from the EPA list of priority pollutants, to kind of ask this question: well, does it still hold when you start to look at a large number of different chemicals? And we've just started to get the results from this analysis, and I'll show that. Here, again, these are the genes on this side. We're now using a much larger gene set with about 15,000 genes. And these are the chemicals across here. And again, we were very, gratified to find that each chemical really has a specific expression profile. Relatively closely related chemicals tend to cluster together. But I think it's very clear that from what we know of the mode of action of many of these chemicals, that that's not necessarily reflected in the mode of action in Daphnia. So they're going to have potentially unique modes of action in Daphnia. So what does this information provide to you, and why was this relevant to screening for green chemistry, the green chemistry initiatives, and understanding the effects of unknown chemicals? Well, and I think this is similar to what Dr. Ankley referred to, is that when you have an unknown chemical, what this sort of approach allows you to do is to try to get some sense of where does an unknown chemical fit into this chemical space. If we have information about what these chemicals do, what their potential adverse outcome is, or other information about them, we can use a relatively simple assay, which is a microarray, which can take on the order of a day or two, and get information about the potential mode of action. And what does that tell us? Well, it tells us we should be concerned, potentially, if it looks like, let's say, lambda silathrin. If the expression looks like lambda silathrin, can't pronounce that, then you should be concerned and look at endpoints that you know are related to that chemical. So it gives you a way to focus your follow-up study based on how the organism is responding to that chemical. So what do we think that this can do? Well, I think one of the things is that it gives us an unbiased screen. So it's not looking at any particular endpoint. So I think that's one of the differences between the genomic approaches, is that we're not going in with any preconceptions about what the mode of action should be. We're letting the organism tell us how it's being affected, and we use that information to try to come up with hypotheses, things that we can test. And that allows us to identify unexpected outcomes. I certainly did not expect that zinc would affect chitinase activity. We found that copper affected immunity in Daphnia. We had no clue to that. And I think there are many examples of this where this sort of work helps you direct your further study onto the endpoints that the organisms are telling you that it's being affected. Okay? And we can potentially define these pathways, and as I was saying, to come up with the appropriate in vitro assays. And then once we've done this, prioritize the chemicals and try to decide which of these ones, based on the gene expression profile, are ones that we should be concerned about. So now I'm going to turn to the in vitro assays. And there's actually a long history of using in vitro assays in ecotox. And there are multiple fish lines that are out there. There aren't many cell lines for other species. And there's been quite a lot of work on in vitro cytotoxicity studies. But they're primarily just cell death, and there are not other assays similar to the ToxCast, which I'll talk about in a second. As mentioned, the primary assays that we have for fish are primarily based around toxicity, the acute toxicity test, for instance, at 96 hours. This is showing that there are a large number of fish cell lines that are already currently available that could be used in vitro assays. This is just an example of one of them, the RT gill cell, which is from the rainbow trout. It grows quite nicely, and you can grow it even in a situation where it will form tight epithelial junctions. And this is used as a model of the gill, and that's an important site of toxicity for fish, because that's where the toxicants can expose it. So there's been quite a lot of work, and this is just showing some of that there have been a whole series of tests here with different fish cell lines, comparing them to human cell lines for a large number of chemicals. There was actually a study many years ago called the Multi-Center Evaluation of In Vitro Cytotoxicity Testing, where they looked at a whole series of different chemicals and compared both individual chemicals to each other, and they also compared them to in vitro versus in vivo effects. And I think, as you can see by some of the R values here, that many of these in vitro tests were quite useful and predictive of the in vivo effects. So there are those. There's a history of tests in the ecotox field and in the mammalian field, as alluded to. The ToxCast program has been developed, and I'll go through this quickly because it's been talked about. And there's a whole series of different assays that have been developed, 624 different assays for each target. And this is showing some of the data that you get from that. So this is, again, a heat map from the paper on this. These are all the assays along this thing. This is the chemicals on here. And you get very similar to what you see with the microarray data, is that you get an expression pattern, and each chemical will give you a different pattern. And again, you can use this information as a way to understand the potential mode of action or mechanism of these compounds. So what's the future now for cell toxicity testing? So in the ToxCast, they've been using primarily single assays. And the next step is to use what's called these high content screening approaches, where you can actually assay multiple different things at the same time and follow individual cells and follow endpoints in individual cells for multiple parameters at the same time. And this has actually been called it's called high content screening. And the hope of with this is that by actually measuring multiple things in the same cell, you can actually get more insight into the mode of action, because you can follow what's happening in a cell or a population of cells over time and see that there's a correlation between all the various endpoints that you're measuring, and help you understand what's actually happening in that cell or those cells over time, and come up with a more detailed mode of action. There are just an enormous number of assays that are currently out there, so you can choose a lot of different endpoints. Many of these endpoints could be potentially relevant to ecotoxicity. But what do we need in order to actually use this approach for ecotoxicity? Well, so for the mammalian system, as we saw from Dr. Ankley's talk, we've got a good start to applying some of these high throughput screening tools to mammalian systems. It has not really been applied to ecological or ecotoxicology. So what would be required to do that? Well, I think we need to identify the cell lines. We've got primarily fish cell lines now. We don't have other species represented. We don't really know what the endpoints are that we should be measuring in ecotox, so we need to define what those endpoints are. We need to develop the assays in order to do that and validate them versus in vivo data, and then ultimately develop specific assays that we're interested in, so that we can actually carry out what I would call an ecoToxCast to help us have a similar data set as they have for mammalian systems. This is just an example from our lab showing the application of that. Can't really see the colors. I can see them fine on my slide. Where you actually we're looking at a whole variety of different endpoints here, and the toxicity of different chemicals and compounds here, nanowires, for example, here. And you can get then information here about a whole variety of different cells. It gives you much better data, and you can see the effect on these fathead minnow cells. So, I guess to try to summarize, well, what is the potential future for ecotoxicology? And I guess the the hope is that maybe we can turn to having an in silico and an in vitro approach in contrast to what is the current method, which is the in vivo approach. Maybe we can develop more mode of action based assessment and develop targeted assays. Try to be comprehensive in our endpoints beyond the standard acute and chronic testing methods. Use high throughput methods such as the high content screening or genomic approaches to help us actually prioritize what we need to do further testing on using perhaps tiered in vivo approaches. Just like to thank the people who've done the work at UC Berkeley and the funding agencies, which went by quickly. And thank you.
I'm interested in the study of the 26 chemicals that you did on Daphnia.
Uh-huh.
And I'm wondering, I think that's especially great because you followed up with the in vivo testing.
Mm-hmm.
And I'm wondering if there might be a way, or if this might have been done, to compare those responses to what's in ToxCast, to compare with mammalian responses, possibly when it is completed, to compare with fish responses, and start to see if there are separate modes of action in between these groups of animals, if they're consistently different, and if we could predict in that way and maybe save some more testing.
Right, so the use of this in sort of cross-species extrapolation. We haven't done that yet for the Daphnia. One of the similar studies that we're involved with was looking at some of those other chemicals I mentioned previously, RDX and 2,4-DNT. RDX is a neurotoxicant, and in a project with Ed Perkins' group, actually looked at the effect of that chemical on fathead minnow, on Daphnia, on quail, on rodents, and actually what was interesting for that, at least for that chemical, is that there was quite a lot of conservation, at least in the basic cellular mechanisms, all the way from Daphnia through to the mammalian systems.
What lab was that?
Ed Perkins' lab. He's part of the collaboration on the fathead minnow data.
Vicksburg Corps of Engineers.
Yeah.
Okay.
Could you guys identify yourself?
Oh, yeah, Dave Appletz, Stanford. So I was very interested in the question of homeostatic response. So, and I wonder whether you're, to me it brings up the question of whether these in silico and genomic responses will be valid, because, and so the question really is, if you have a homeostatic response, is the organism really normal, or is there some stress? And I'm not convinced that you can get the stress in a single organism assay. It has to be done in an mesocosmic or some sort of competition situation. Is that something that is that you would consider or is being considered?
So the question is, one question would be, how do you distinguish a homeostatic response versus a response that ultimately leads to an adverse outcome, which is potentially, and I think that's where you really have to do this sort of correlative studies, where you are correlating what's happening on the gene expression level with other indicators, such as physiologic endpoints, tissue damage, other sorts of approaches. I don't think you're necessarily going to be able to get that from this sort of genomic approach initially. You're going to have to, the term is called phenotypic anchoring. You're going to have to sort of, at least for some chemicals, you're going to have to be able to show a direct linkage between the measured effect and your endpoint of interest. And I agree with that The hope, of course, is that you're not necessarily going to have to do that for every chemical, because then you haven't saved yourself any time. But once maybe you've identified a limited set of adverse outcomes, then you can define those linkages between something you can measure in the organism in a relatively rapid way, such as gene expression, and show that that is, in general, linked to a physiologic response and an adverse outcome.
So my question, I guess, my question really is whether you can assess adverse outcomes, whether a tissue response or that sort is really an adverse outcome if you, because shouldn't you go, or maybe this is done, it's my ignorance, but go into a competitive situation. How is the organism surviving with its normal competitors under very low levels?
That's a problem even with standard toxicity assays, right? In that you're generally just looking at that organism in isolation, and you're not necessarily seeing how it's interacting with its environment. And I think that's a general concern about these approaches. I mean, ideally, a mesocosm study would probably be the best way to do it. The problem is that adds another order of magnitude of complexity, as well as cost. But it is an interesting point, and that perhaps once we define, and we're still a long ways from that, a limited number of pathways which are ending up in adverse outcome, then maybe we can go to the next level and actually see how those changes actually result in behavioral effects or other effects in a mesocosm or in a more real world situation. I think that's the real hope, is that if you're facing an innumerable number of chemicals, you can't do this. But if you can narrow this down to a set of definable pathways that occur for multiple different, then we can do the more focused studies and tie that to the ecologically relevant endpoints.